In this part of the lecture, we're going to start talking about translation, which is the process of using the messenger RNA that we created during transcription as the instructions to put our amino acids together in the correct order to create a protein. So translation, the RNA-directed synthesis of proteins, will happen in the cytoplasm or the rough ER, but it always happens at a ribosome. And ribosomes, remember, can either be free in the cytoplasm or they are embedded in the rough endoplasmic reticulum. And in this process, we're going to need several different things. We are definitely going to need some ribosomes. We're going to need some RNA molecules called transfer RNA or tRNA, and we're also going to need some messenger RNA, which is what we made during transcription in the nucleus. The messenger RNA is going to be crucial in this process because it actually has the information of which amino acids are in the protein and what order they go in. And that information is stored in short 3 nucleotide sequences called codons. And those codons are kind of like a secret language. They're like a secret word that means a particular amino acid. So for example, when we're looking at our transcript here, this is our messenger RNA at the bottom of this slide. At the very beginning, we have adenine, uracil, guanine. It's a 3 nucleotide sequence, and that is a codon. To figure out what each codon means, we need the secret code, which is going to be the codon chart. So in our example here, AUG is our codon. So we have to look at the first letter first, which is adenine, and we're going to start on the left-hand side of our chart. And we look for adenine. So we've got uracil, cytosine, adenine. So we know that we are somewhere in this row. Okay. The next codon is uracil. So then we go to the top of our chart and we look for U, which happens to be what we're looking for. So we know that we are in that column. So we're somewhere in this box here. And then finally, we look at the last codon, which in this case is a guanine. So we go to the right side of our chart and we look at uracil, cytosine, adenine, guanine. So we know that we are the last codon in this chart. And that codon will translate into the amino acid methionine, which is abbreviated here as MET. Then we would continue reading our messenger RNA and look at the next 3 nucleotides, and that would be our next codon. So in this case, we have AUC. So if we use our codon chart, we see that we're going to be in this 3rd row here. We're going to be in the 1st column, but this time we're right here. And so AUC stands for, or it means, isoleucine in amino acid language. So for our protein so far, we would have methionine peptide bonded to isoleucine. Our next codon is UCG. So again, we'd look for the uracil on the far left, so we know we're in the top row. The cytosine on the top, so we know we're in the 2nd column, and then we just look for the guanine as our 3rd nucleotide. And so UCG, that codon, really translates into the amino acid serine, which here is abbreviated as SER. And then in our last codon, we have UAA. And so if we use our codon chart here, we see that UAA actually gives us a stop signal. So we have one start codon, which is AUG, and it will always be AUG. And then we have 3 stop codons, UAA, UAG, and UGA. So the messenger RNA is storing all of the information about which amino acids are in the protein and what order those amino acids go in, but something is going to have to actually bring those amino acids to the ribosomes in the correct order, and that is the transfer RNAs, or the tRNAs. Their job is to transfer amino acids that are free in the cytoplasm to the ribosomes. So these are RNA molecules that are about 80 to 100 nucleotides long. They have a tendency to fold over on themselves and hydrogen bond with themselves, and that gives them this characteristic T shape. At one end of our RNA, we have this purple amino acid, and that purple amino acid is going to be very specific to the RNA. It will only carry one type of the 20 amino acids. At the other end of this molecule, we can see a region called the anticodon. So that's here. These are the nucleotides that are exposed at the bottom. Now remember, the whole thing is made of nucleotides, but these are special. These are the ones that are going to hydrogen bond with the codons of the messenger RNA. So they are going to be complementary to the codon, hence anticodon. So in this particular case, we have uracil guanine guanine, which would be complementary to the codon adenine cytosine cytosine. ACC, go Riverbats. So if there was a messenger RNA transcript that had the codon ACC, this particular transfer RNA with the anticodon UGG would be attracted to it, and it would bring the amino acid that codon encodes for, which in this case is threonine. So one thing I want to point out with this codon chart is that we do have redundancy built into it. So if we were to look at a very specific amino acid, let's say alanine at the bottom here, we have 4 different codons that encode for alanine, GCU, GCC, GCA, and GCG, which means that we're going to have 4 different transfer RNAs that would be complementary to those codons that would all carry alanine and only alanine. Okay, so let's get this process started. So on the left-hand side of the screen, we can see all of the things that we're going to need. So we have our RNA transcript, our messenger RNA at the bottom there that's containing all of the instructions in the form of codons. We have our tRNA at the top here, which is going to be transporting amino acids to the ribosome. And then we have our large ribosomal subunit and the small ribosomal subunit. And each of these are made in the nucleolus of the nucleus, and they are a complex of ribosomal RNA, or rRNA, and proteins, which have just folded just so that we get a small unit and a large unit. They're made separately in the nucleolus. They leave the nucleus separately, and then they'll come together during this process. So the first thing that needs to happen is we're going to have our messenger RNA orient itself so that it is 5' to 3' on the small ribosomal subunit. While that's happening, our initiator tRNA is going to bind with our start codon. So remember that our start codon is always adenine, uracil, guanine when we're reading it 5 to 3. So it's always AUG, which means that the transfer RNA that would be attracted to AUG would have the anticodon, which would be UAC. And then attached to that transfer RNA, we have this amino acid, methionine. Once that is all bound, we have our small ribosomal subunit, our messenger RNA, and our initiator tRNA attached to the start codon. That is when our large ribosomal subunit bounces in just like that, such that our transfer RNA is in the P site of the large ribosomal subunit. The next step is elongation. So all we need to do here is read the next 3 nucleotides in our messenger RNA. So read our next codon, and then let the transfer RNA that has the anticodon come in, bringing the amino acid with it. So here we see the transfer RNA that had the anticodon that was complementary to the messenger RNA codon has entered the ribosome at the A site of the large subunit. Then we can see this chain of amino acids, all of those purple shapes that are connected to each other by peptide bonds, is going to be transferred from the middle tRNA, the tRNA that's in the P site, over to the diamond-shaped amino acid that's attached to the tRNA in the A site. So in this slide, we see that successful transfer of the polypeptide chain over to the tRNA that is in the A site. Then everything will shift over. So that transfer RNA that was in the P site will then go into the E site for exit, and it will exit the whole complex and go pick up some more amino acids of whatever it was carrying. The messenger RNA moves with it, which means so does that transfer RNA that was attached at the A site. And then that will open up the A site again so that we can continue reading the next 3 nucleotide sequence in our messenger RNA. Our transfer RNA with the anticodon that's complementary will attach. Then we transfer that chain of proteins over and the cycle continues. And so this will continue until we get to a stop codon, either UAG, UAA, or UGA. And when we get to one of those stop codons, that will initiate termination of this process. So there is no transfer RNA that has the anticodon to these stop codons. Instead, a molecule called release factor will attach at the A site with the stop codon. Once release factor attaches, it's going to cause the entire complex to dissociate. So the transfer RNA is released, it will go pick up more amino acids. Our messenger RNA is released, our subunits, the large and the small ribosomal subunits, split apart from each other, and we have our free polypeptide. And all of these things can be used again: the ribosome, the messenger RNA, the release factor, the tRNA, and then we have this free polypeptide. Now, sometimes a cell will need to make a lot of protein really quickly in response to their environment. So we might have one messenger RNA and several ribosomes reading it all at the same time. And we call this polyribosome. So that's what we see in this picture, where that red line is the messenger RNA, and we have 4 different ribosomes that are translating that messenger RNA into the purple polypeptide in this case. And that's how cells can make a lot of protein really quickly if they need to. Once we've made those proteins, then all they have to do is go through their regular protein folding, creating the secondary structure, the tertiary structure, and potentially the quaternary structure. Usually they will do this on their own, but sometimes they might need some help from specialized proteins like chaperonins, or they might be sent to the Golgi apparatus where they'll go through modification and then they will be shipped off to wherever they need to go in the cell or the extracellular matrix. So here we see a summary of everything that we've talked about in terms of gene expression. So in the nucleus at the top here, we have our DNA being used as a template to form the RNA transcript. So we see that teardrop-shaped protein, which is RNA polymerase, that is assembling that messenger RNA. Then that messenger RNA has to be processed. So we add that poly A tail, we remove the introns, we add the 5' cap, and then it's able to leave the nucleus and go into the cytoplasm. Once it's in the cytoplasm, it will associate with a small ribosomal subunit, the transfer RNA, and the large ribosomal subunit, so that it can be translated into a polypeptide chain. So this would be a good time to pause and go watch that animation. Actually, I posted 2 animations about this process.